So now we're coming to museum collections. So museum collections is not a storeroom. I have to emphasize this because there are some people who think like that. No, it is not. It is important references. And mind you, some type specimens, they can actually be very expensive. And those are one of the driving values for our museum collections. Okay. So if you have a collection or museum or herbaria, you have different kinds, which is like local collections. If you're working on botany, you have botanical gardens, or you have museum and herbarias. Okay, so let's try to look at some of these. So, with regards to local collections, okay, so perhaps in your biodiversity lab, we have some specimens that were collected from the field. So I'm trying to relate as best as possible with you guys. So of course, you guys have done work from Bidong Island. So all of those samples, they might actually keep some samples in their lab for their collections. But some will have other places like agricultural research stations. And then you have state geological surveys. Okay, so those who are doing geology, they will actually have their own collections of minerals. And then you might actually have a marine laboratory. Again, a field station will be more like, say, Bidong Island. Marine laboratory will be more like the biodiversity lab. Okay. Now, although this can be helpful collections, the issues are they're not always well maintained. Okay. Sometimes those who maintain it are research assistants or available investigators. Okay. And you don't want those kind of things because you want specimens that are well maintained and they are registered, and it's not just being registered locally, it's such that it's internationally recognized. So if you notice how Azwarina and team have ensured that the collections in RRC are established to, say, MyBis, to any other zoological museums, to natural history museums, and even the fact that we can loan specimens internationally, that is the establishment that you want to get to. If it's just for the sake of field stations, etc., so it will only be within, not even on a national basis, it's just only within, say, within UMT that might know about these collections. Again, it can be helpful, say, when you're doing field studies, like you're taking your kerja lapangan work, you're doing marine invertebrates, you just want to learn about this, it's fine. But if you want to do extensive work, it may not be very helpful. Okay, so then you have botanical gardens. Now, botanical gardens, they actually have plant species, and that, of course, they have plant species, and scientists, they collaborate with scientists to look into other aspects such as reproduction, phonetics, variation, and also hybridization. Okay, but then you also have herbaria. Do you know, herbaria also holds plant collections. But what is the difference between botanical gardens and herbaria? Now, the interesting thing is botanical gardens is they have live specimens. That's the difference. So that's the keyword. That's why the scientists are able to look into its reproduction, its phonetics, the different variations of the plants, and whether you can make hybrids. Okay, but a herbaria, of course, will only be research collections of dried plants as such. So can you imagine? I kind of forgot which museum picture is this, but this is their collection of herbaria. Isn't that amazing? How they have painstakingly kept each one of these dried specimens so well. These are the kind of things we hope at least RRC can come to one day. But anyhow, so if you're talking about museums, it's a building in which objects of historical, scientific, artistic, or cultural interest are stored and exhibited. So mind you, when it comes to museums, there are different kinds of museums. I haven't extensively looked into this yet, but because we have a speaker from Singapore who will actually be looking into this, so she will explain to you in detail. But generally, museums, depending on what you're looking at, if you're talking about, say, plants and animals, that is what you mean by a natural history museum. Okay, but if you're looking into historical museums, of course you're looking into back in history. So you will have, so example, like the history of Malaysia. Okay, so you will look into things like all the stuffs and equipments back in World War I and World War II, or times of Paramiswara, that kind of thing. So this is historical, and you will have all these batu-batu nisan and stuff. So that's historical. Scientific, it really depends. It's very broad term. Whether or not you're looking into a scientific museum will be something like our Pusat Sains Negara. So it looks into different forms of research or methods and things related to science. Okay, and then you have the artistic museum. Of course, it's solely based on arts, and the way it's exhibited is also very different compared to a natural history museum. You have those of cultural interests. Our museum Terengganu very much caters into cultural interests, so a lot of their money is pumped into looking after the batiks, etc. So, but we have a small room for natural history technically, so that's why it's not really a natural history museum. It's more of a cultural museum in Terengganu. Okay, so they are stored and exhibited. Exhibition and storage is very different. So if you notice, for the lab work that you will be doing, the process of what we're explaining to you now is a lot of what you're doing based in a stored environment. Okay, how you're preserving, how you're fixing the specimens, how you're ensuring the collections are conserved. Again, conservation of the wild is very different from conservation of specimen collection, because you have to ensure that there's no fungus, it's well managed, it's not damaged. If you have to change the ethanol, those are things you have to do. The exhibition is what you see in the front. Okay, and this is the finale of your lab work, but what you guys will be doing is the virtual exhibition. So there's a difference. Okay, we've covered herbaria. And so the objectives of this work are providing preservation and conservation, and providing systematic research. So in my case, I actually had the opportunity to be based in the Museum of Tropical Queensland. So they had one of the biggest coral collections in Queensland, where the Great Barrier Reef is. And then I also got to be based in Sydney Museum. So in terms of research, it is important to have such museums, because it is people like us who will actually continue to form attachments with them and actually work on the collections. Okay, and then of course you have university, state, national, and private, which is non-profit, which houses more than 60 phyla. So what I mean by the state, university, state, national, private, and private non-profit is all of these stakeholders that actually would want to use museum collections. Okay, and of course there are those museums that actually are more advanced, and they have electric cataloging right now, more things that are done through online, etc. So okay, this is what you guys may have seen more often not. This is museum exhibitions, which is solely catered for educational purposes. Sometimes they can have some form of festival, but ultimately exhibitions are usually done for educational purposes. So the layout of for most museums is such that you will go to the ticket counters. So that money is actually a small portion of the money that actually helps with maintenance of the museums, or to conduct some of their scientific outreach programs. Okay, and then of course they also do have a souvenir shop sometimes, and of course you have the section where they have the exhibition. So they will have the educational side, you can take your pictures, etc. So this is what you see or what we use as the front of house. Okay, but this is where the true magic happens, where we call it the back of house. Okay, and yes, if you actually get to work in such museums, it's amazing to work in such museums. Okay, so this one obviously has a broad range of avian collections. Okay, that it's extending right up to the end here, so it's amazing. And then, of course, you have those who look into fossils. So you notice how, like, I put the picture here for herbariums, how the storage is quite different from um, how, say, birds are kept. So they're kept in extensive drawers like such. For fossils, you have smaller drawers. And then, of course, those that are the wet collections. Compared to these are dry collections. So the wet collections are such. These are exposed cabinets, but some will actually have cabinets where you can actually roll the cabinets. So these are the kind of things that we have. So in terms of how RRC is operated, their web collection such looks like this, almost pretty much like this. Okay. So now you might think, oh my gosh, how are you going to look at all this material? Like where do you start? It's mind-boggling. Okay, so when you're locating the material, of course, it's best to first Google, email, and ask or meet an expert, or even with the curator. So usually it's going to be either one of these. Either they will revert you to the curator, the curator will revert you to the expert. Okay. And then when you're emailing either the expert or curator, you want to help them by at least providing them with the modern description of the journal. Okay. So don't go emailing your experts and go like, hey, I'm actually interested in looking for this Sardinella fish. Can you help me? I mean, well, yes, of course they want to help you, but you need to be a little bit more professional, and you have to explain what is the issue about. Okay, you want to find the type material because we found this particular species here, but we're not too sure if it's a different species, etc. This is the best available journal that I found. So again, it may not necessarily be the modern paper, it may even be the old paper that you're talking about. Okay, so because the expert or the curator would want to know which paper are you referring to if you want to look at their collection. So example, you are working on a specimen from Malaysia, but then you found this specimen, when you looked at the paper, it's from Japan. So you want to locate that particular specimen back in the museum in, say, in Japan. So you will have to at least help the curator or the expert provide that particular description. Okay, so this description may include lists of institute types, and non-types are deposited. So you have to provide the accession, catalog number, locality, collector, and holding institution. Okay, I will give you an example later from the Natural History Museum for France, and we'll play around just so that you have an idea how what you're looking for. And again, older papers may not all provide this information. Again, but don't entirely exclude them. If you happen to find modern papers, this is great. Older papers can help, but again, it's true, it may not provide detailed information because specimens may no longer exist. Some of this happens, example, like say, if an old museum was established back in where World War I, World War II happened, okay? And because of that, the buildings were actually extinguished, so much of the specimens were gone. So these things are also likely to happen. And then incorporated into museum collection and can be located through directories, okay? So basically sometimes some of these papers actually have those missing details. So it is true. So sometimes again you want to ask an expert, and you may say, is a lot of these situations have happened in a lot of all of these other papers from experts based in Europe. So that's why one has to assume that it's going to be either found in, say, in a museum in Sweden, or in a museum in in France, etc. That kind of thing. So sometimes museums are insufficiently funded to make all these collections available. This is a known fact, okay? Why I really want to emphasize on this is because I think a lot of people don't seem to appreciate or understand the functions and its purpose of museums. We're not just a storing center for specimens, we actually hold a lot of extensive important information, but it's, again, we are underappreciated, and that's why we need to be made known of the kind of work we do. Okay, so then if you're able to go to the museum in Japan itself, example, it's nice and wonderful. But now in this case that you don't have enough funds and you want to borrow the material, so you have to do a written request to be shipped, um, again, or during a visit, whichever the case, you have to have a written request. So obviously you have to deal with the curator because they will provide you with the museum loan policies. Some can be very easy, some can be very songeh. All right, mafan, very difficult. So, and then it must be stored and shipped under conditions that meet or exceed the home institute. So, which is why I've mentioned that if you were to do borrowing of material, say from specimens for our biodiversity lab, it's not definitely not going to meet into that standards. But say with RRC, we are actually certified. We actually have the certificate to do those kind of shipping. So that is why you have to ensure that those museums do have such certificates. And this is very important. Any intentions of dissecting requires approval, especially if you're working on the type material. Because don't forget, it's not just you who's going to be using it. Others would actually want to use and check on it. By you dissecting it, you're actually going to destroy it. So we are very, very particular about dissecting any particular specimens for that matter. Okay? So, again, a biologist, a taxonomist usually is a biologist, but a biologist is not necessarily a taxonomist. So remember how I mentioned earlier about how it, taxonomists are actually quite picky people, but for particular reasons, okay? And then, of course, now as a taxonomist, or at least learning taxonomy, you would have understood that because a species has differences in terms of its biology, its ecology, even paleontology. So the kind of species concept that you're dealing with is actually very different. Okay. So with taxonomy is a specimen. So with taxonomy, okay, what we hold as an important hypothesis is the specimen itself. So the specimen serves as a reference standard for taxon and is used in references for hypothesis. Okay. For us, the actual specimen that you're dealing with, especially if it's a new species, that is what you call the type. All right. Now, there are 4 different types in the zoological nomenclature, and we call it the name-bearing type. Okay, I want you to really pay attention to this because I may ask this in the exam, so you really need to get this right. Again, for taxonomy, in terms of our hypothesis, is the specimen that is used as the reference standard, okay, our reference form. Meaning to say, if there are many taxonomists who want to debate about a matter, we have to go back to that specimen that you can see with your naked eyes. Okay, but there are different kinds. There is the name-bearing kind and the non-name-bearing kind. So with the name-bearing type, the name-bearing type is the biological type that determines the application of a name under the International Code of Zoological Nomenclature. I will cover one topic specifically for this, but just for the sake of this lecture, please understand that when one is naming a holotype, a paratype, a syntype, whatever type it is, we have to go back to the ICZN. But this is only for zoology, yes. If it's for botany, it's a different code altogether. So please bear that in mind. Anything to do with zoology, whether it's marine, aquatic, even terrestrial, we refer to International Code of Zoological Nomenclature. Okay, so I've mentioned there are 4 name-bearing types. Okay, the 4 name-bearing types that you must remember is first, the holotype. Okay, the first one. So the holotype is a single specimen. A single specimen, okay, it cannot be more than one. Used by an author, the author who is about to name it as a new species, either the only specimen he found or one of several found. Okay, example, again, but the only one designated as a type. So example, imagine yourself coming from Pulau Bidong. You found 10 fishes. Okay, example, you found 10 fishes. So from this 10, you realize, hey, I've checked this out on the literature. I've checked this out on the collections. I have not seen this species ever before. Definitely going to be new. Okay, so if you've determined it's new, and the thing is, if it's only one that you found, it's very easy. That is a single holotype. As I mentioned, only specimen he found. However, it is one of several, meaning to say you found 10. It is your decision to choose out of this 10 which is the one that should be designated as a type, because it's only that one that will be a holotype. The rest of the 9 fishes will be considered as paratype. I repeat, the only one that you find from the 10 is called a holotype. The rest is called a paratype. The term paratype is not a name-bearing type, okay? It's just a type, meaning to say it's replicates of it, but it's not the name-bearing type. Now this, when I mean by name-bearing type, is actually very important compared to non-bearing type, because meaning to say, if taxonomists were to look at references, we preferably look at the non-bearing type, of the name-bearing type. So holotype is one of those name-bearing types. Okay. The second one is designating a neotype, and that is as a specimen selected and designated in a paper in literature. Meaning to say, if a paper actually, example, if you lose this holotype, for example, but you have a paper with a picture of the holotype and it's plentiful, even the picture in a publication can be considered a holotype. However, if the picture is one that is not clear, example, like I showed you before in this picture, okay? Okay, example, this is the holotype, okay? But you can't find the specimen. If it's easy to recognize, it's fine. You can leave it as it is. However, say if it's a holotype that is found in 17 and 1800s, and this is all you have to work with, plus the description is in Latin. So it's not of modern taxonomic standards. This is a time that you would actually have to designate a neotype. But mind you, designating a neotype can be very, 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 very tricky. Not every taxonomist is going to easily replace neotype if it's damaged, lost, or destroyed. Okay, they will find every form of reason to ensure that there's a way to that there's no neotype designation. Only under very strict circumstances where it's totally lost, destroyed, or cases like this, there's no other replicates of any specimen, and looking at this picture is hopeless, then can you designate a neotype. Okay. Are we clear with this too before I move on to the next? Any questions?
Yeah, doctor.
Good, yeah? Okay. Okay, sorry, I need to go into this part first, yeah? Okay, the third form of name-bearing type is a syntype. Syntypes these are 2 or more specimens selected from available material to serve as types. Okay, syntypes usually have occurred back in like the 1700s, 1800s, etc., where a lot of taxonomists have designated specimens as types. However, in the description, they don't specifically indicate which of the example in the species that they found. Say example, you have okay, let's use a different example here. Say in the 1800s, a former author by the name again of Minot, say he found 8 different kinds of crustaceans. 8 crustaceans of species A. Now the thing was that he described it, but he did not designate a holotype from this 8. That means all of these 8 are just types. So this is going to be tricky because, as you all know, now in modern taxonomic standards, you have to have one holotype. But a lot of taxonomics back then, again, taxonomy was still ever evolving. So they didn't quite designate out of the 8 which of the one that he should choose. So if you were to come and work on his work again, what you would have to do is you would have to pick out one of the 8. Now, how do you do this is first, you have to look back at Minut's paper and you have to understand the description as best as possible. So one tip that you have to look out for is perhaps the length. Yes, of course, when you preserve these specimens over time, it is bound to have shrunk. It is bound to have some form of metabolic change over time. Okay, but if it's easy, if it's easy, like to example, say if he was describing from an adult, and just by chance there's only 2 adults and the 6 are all juveniles, then it's going to be likely one of the adults. But if say all were adults, then you would actually have to make the decision, if in terms of decision of choosing one of these 8 syntypes as a lactotype. Okay, so again, syntype, the scenario here is that there's 2 or more, like say in Minut's case, he had 8 specimens, but he did not allocate a holotype. So all of these 8 specimens are syntypes. Now in a case you want to work on Minut's work and you want to designate, like say, a holotype for this. However, because Minut has already talked about this, you cannot take credit and say and work on it as a holotype because he already described it. But what you can do to designate one of the 8, which is equivalent to holotype, is called a lactotype. Okay, so a lactotype is one specimen of the syntype sets, which is one or more specimens known to have been used by the author if no type was identified, may be selected as the lactotype by the author of a later worker. So I hope you understand what I mean. Example, in the 1800s, there's one guy by the name of Minut who worked on 8 crustacean of species A, but he did not identify any one of the 8 as a holotype. So all of these 8 are called syntypes. In the case of lactotype, one of you decided to work on Minut specimens, and then you choose, okay, based on the description or based on your best identification on how the species is identified, either it's through a female or male. Okay, you will have to be the expert to identify that. When you are choosing one of the 8, it becomes a lactotype. Okay, so lactotype, as I mentioned, one specimen of the syntype set and may be selected as a lactotype by a later worker. But just by you selecting doesn't mean it ends there. You would have to publish the paper also. Then it should be the specimen illustrated in the original description. So this is what I mean. If at all the author has illustrated it in the original description, has given a description of the length, etc., then of course that's how you choose. But if it's not depicted, then you will have to make the best decision. So remember how now you've chosen a lactotype. So what becomes of the balance 7? So then the balance 7 will become a paralactotype, which is an additional specimen from among a set of syntypes after a lactotype has been designated from among them. So, but if a holotype, the one that you are identifying directly, because now in modern standards we know how to do. So if you have the 10 fishes example I was talking about, one is considered a holotype. The rest will become a paratype. So paratypes are specimens that the person making the original description examined while carrying out the work. They may be from the same or different locality, but they clearly, at least in the mind of the describer, are members of the new species. Okay, to the best of your ability, you realize that they are the same species. Okay, although they are not types in the nomenclature sense. Okay, so this is what I mean by the non-bearing type. You notice how all the ones in red are the ones I put as bearing name. So the ones in blue are the non-name bearing types. Okay, so although they are not types in the nomenclature sense, they are often distributed to other museums as vouchers for the new species. So museums will only hold the main important one. The rest, if you have extras, you can distribute to other museums. So like holotypes and syntypes, they are often catalogued and stored separately from the rest of the collection. So all the bearing types should be in one collection. All the extras, the replicates, will be in another form of collection. So you notice how this is termed as paratype for holotype. So in order for you to remember lectotype, and the balance will be just add on the word para, lectotype. It's parallel to this. Okay, parallel in the sense of the location, the time, etc. Now I have a question for you. Just say, you found the Sardinella, the 10 species, okay, you describe the 10 species, you found it in Bidong in year 2020, and you describe this paper. Now, if you were to collect the same species in a later year, say 2021, does it hold any type of type, any type bearing name? Will it become a paratype? Does it become a holotype? What does it become? Any idea?
Another paratype?
Wrong. It becomes just material examined. So only at the time when you found and when you described your paper at that time, those are what it holds, holotype and paratype. The next time you find, although it's the same location, same coordinate, everything's the same except a different date, it just becomes material examined. It doesn't even hold any non-name bearing type. That is what you need to understand, yeah? Even though you are the one who first discovered it. Okay. If anybody else were to find also, it's the same thing. If some of your friends were to find the same species, etc., from that location, it doesn't hold any kind of type status, not even paratype. It just becomes material examined. That is what you need to understand. Okay? Are we clear? How is everyone faring? Any questions? Because usually I
Yes.
always get questions here. All right. So I assume that none of you have any questions. Okay. You actually have the term topotype. These are specimens collected from the same locality as the type material, although not necessarily at the same time. Okay. So there are times that you, if it's from the same locality, you can actually use the term topotype. Okay. So there is no official standing, as I mentioned. So that's why some even use the term material examined. But if you need to put an official standing, you can use at the term topotype. But if a museum has such material, it is usually well worth seeing. Okay? Well worth seeing in a sense of extra material, and you can hold a topotype status. But however, again, sometimes it really has no official standing, so much so people actually call it material examined. That's all. So Winston in 1999 vouches that the fact that with marine invertebrates, topotypical material of the species 1/6 is much more likely to belong to that species than material labeled with the same name but from thousands of miles away. Yeah, so with topotype, it has a better standing ground if it's from the same coordinates, from the same locality, rather than from miles away. Okay, so when you're visiting collections, what are you to expect? Remember, yes, I did mention about you having to write to the curators, etc. But now you are at the place. So what you're looking at, yes, although you want to look at the species you're looking at, look at all species from the same genus. Look at all different kinds of species from the same genus preferably, or even the same family. Especially changes with the taxon not stored under current names. Okay, there are some incidences where probably they can only identify even to class or family. Get a hold of all those registrations as well, because there's a likelihood that you will be able to help some of the curators. And then check for any catalog or possible misidentified specimens. This has happened many times also. So when I've done my collections, I've actually looked at the whole lot of specimens, about like nearly 200 to 300 different specimens with the same, under the same name. But when looking through some of them, I actually managed to find those that are two of different species actually. So sometimes things like this can happen, but it's to the best knowledge of the curator or experts there. But if you are one who's going to write a paper, you are in a better position to actually help these curators, okay? So you may be filled with a half dozen names, and some given accession numbers may never be catalogued, okay? So again, we have a sense of ethics as well when you're cooperating with systematists. So how would you do that? It's supposed to be a win-win this situation, because of course for you are publishing your paper, but you're also establishing the museums and looking out for using their materials so they have a better ground to actually get more funding. So this, you cannot rush taxonomy, because you really have to have an eye on the work that you're looking at. It's like having a momentum for something. Once you work on it, you become better at it day by day. It's a practice, okay? So you need to give some time for the work that you're doing. Even for me, having to read just the general understanding of work, it took me about a good 3 months before I can really start identifying things, okay? And then you have to learn to sort the material yourself. You cannot afford to always expect the curators to be behind your back. You have to work very independently, okay? Don't send an excessive amount of requests or emails for that matter. Just be nice and be cooperative, and call, email, and write ahead of time. Please always be organized. Don't ever do last minute work. It seems like very straightforward things, but sometimes it can be very irritating for curators because or experts sometimes, because they already have so many things on their schedule, and to be very demanding at very limited time, it's not fair to them as well. So you need to actually be very understanding, because they're multitasking a lot of things. So sometimes why I want you to be, if you had a chance, I would really like you to work hands-on with Azwarina and team, so that you have an idea the kind of extensive work that they do. Okay? All right. So I'm coming to the last parts of my work. So let's play around when you're looking for type specimen. Okay? There are most other museums, established museums, okay, say in the US, like the Smithsonian Museum, even in Australia for the matter. So they're on their website, they will have things like this. The visiting part is the visiting hours and looking at the exhibition. But if you want to look at you as a researcher, and you want to look at the research or expertise of your particular field, say if you're working on mollusk, you want to see who is working on mollusk, or if you want to look at their collection, so this is the place that you're looking at. Okay, even with Australian Museum also, they will have similar platforms. Most established museums will have a very user-friendly platform. So I'll just give you an example of how to look for here. All right, and later on you can play around at your own terms. So example, like in my case, I'm very much interested in looking at crustaceans. So if you look into this, you're likely to get all of these. So if you want to do a loan request and consultation, so this is the part you want to email. But before you email, try to make an effort in looking at the collections itself that they have. So of course, look at this here. You have a biological anthropology, cultural anthropology, terrestrial arthropods, botany. So I'm interested in marine invertebrates, okay? So let's look here. So if you were to look here, so what you will see is, okay, sometimes, okay, it gets very tricky because they will write it in French again. But okay, Sorry. Okay, this is good. Just have it in English. And so then they'll show you their collections here. Okay, what they have. Annelids, ascidians, bryozoans, cnidarians. And like, lo and behold, I even have parasitic worms. There you go. Poriferous, protists. But I'm interested, of course, in decapods and non-decapod crustaceans. Okay, so when you're looking at that, then you have to be a bit more detailed. Okay, you can either go to the collection database, and these are the people to contact. So this is how it's very helpful for you. But okay, more specifically, this is a search button. So say example, I'm looking for a species of, at least for this genus. Or perhaps I actually have to go to the collection database. I do apologize. This this is very sad. I was just about to show you what they have in their collections, how to make a search. Just hang on there and we'll see what's up. Oh dear, this is very unfortunate. I do apologize. But basically, what it will have, if I can give you a screenshot, I will try to give you a screenshot later. But it's very helpful in the sense that it would actually give you a checklist of what you can search for. And then if you were to type the name, it will show you whether you have a type specimen or non-type specimen in their collection. So then moving on, if you have a chance to play around, you can play around. Okay, so now for next class, what I would really like to do with you is, okay, I've mentioned here each group. I'm happy for you to actually find it individually, actually, so that you have an idea of what to look out for. So for every person, please bring a soft copy of your taxonomic paper. There are many different platforms. I will show you how to do such searches for taxonomic papers the following time. But for now, I just want you all to go to Journal of Zoo Taxa. And you can verify with me on Tuesday if the paper is okay, next Tuesday. And then find a title representing your organism of interest. Identify the different parts of your taxonomic paper based on today's handouts. And each paper will summarize on how to read their taxonomic paper. Okay, so what you can do is you can also go to WhatsApp and you can actually share with me what kind of papers that you have. And then I will provide a link on eLearn so that you can upload all of your different zoo taxa papers. So that we'll show, then later on that one of the classes, we will actually discuss how to read a taxonomic paper. Okay, again, I do apologize that we are unable to look out for this. It was actually running well when I looked at it in the morning. But things like this do happen. So other than that, do you guys have any other questions? Yes, agreed. Thank you. So are you guys okay with Sunday? Cool. Actually, in fact, you guys can start uploading it even on a Saturday, Max. How is that? Then we can discuss class on Sunday. All right. So, okay, good. So, upload all of this by Saturday, examples of your paper of interest, and then we try to discuss some of these papers. Okay. So that's it from me. If you don't have any questions, thank you very much for today.